palmitic acid yields more ATP than glucose. Reason: R carbons in palmitic acid are more reduced than those in glucose. A. Both A and R are true and R is the correct reason for A. B. Both A and R are true but R is not the correct reason for A. C. A is true but R is false. D. A is false but R is true. Q.29. When cell components are fractionated by sedimentation, the correct order, from lower to higher gravitational force, G, in which the components get separated is blank. A. Nuclei, a population. The probability of a susceptible individual getting infected with SARS-CoV-2 is low when a majority of individuals in the population becomes immune to this virus. This phenomenon is known as blank. A. Innate immunity. B. Adaptive immunity. C. Active immunity. D. Herd immunity. Q.31. Given below are 4 reactions of the glycolytic pathway catalyzed by the enzymes E1, E2, E3, and E4, as indicated. Which of these enzymes are not part of the gluconeogenesis pathway? A. E1, B. E2, C. E3, D. E4. Q.32. Which of the following molecules is a second messengers produced by the phosphoinositide signaling cascade? A. Phosphatidyllinositol 4,5-bisphosphate. B. Inositol 1,4,5-triphosphate. C. Inositol 1,3,5-triphosphate. D. Diacylglycerol. Q.33. A protein has 7 cysteine residues. The maximum number of disulfide bonds of different combinations that can possibly be formed by these 7 cysteine residues is blank in integer. Q.34. A lyophilized intracellular concentrations of ATP, ADP, and inorganic phosphate in 4 cell types are given below. Which one of these cell types has the most negative increment G for ATP hydrolysis? Cell type: ATP MM, ADP MM, inorganic phosphate MM. L 3.0, 1.8, 5.0, K 3.9, 1.3, 3.0, B 2.7, 0.7, 2.7, M 7.2, 0.9, 8.0, A, L, B, K, C, B, D, M. Q.37. Which one of the following amino acids has more than 2 acid-base groups? A. Alanine. B. Leucine. C. Phenylalanine. D. Tyrosine. Q.38. Enzyme activity profiles as a function of time in the absence or presence of different types of feedback mechanisms are shown in the figure below. Match the following feedback mechanisms with the corresponding profiles in the figure. P. No feedback mechanism. Q. Negative feedback mechanism with short delay. R. Negative feedback mechanism with long delay. S. Positive feedback mechanism. A. P. I. Q. E. R. E. S. I. B. P. I. Q. E. R. E. S. I. C. P. I. Q. E. R. E. S. I. D. P. I. Q. E. R. E. S. I. Q.39. A linear DNA fragment of 5 kilobase kb, when completely digested with ECRI, produces 2.5 kilobits, 1.5 kilobits and 1 kilobit fragments. Complete digestion of the same 5 kilobits fragment with XBI produces 3.5 kilobits and 1.5 kilobits fragments. Which one of the following sets of fragments will be obtained if the 5 kilobits fragment is fully digested with ECRI and XBI simultaneously? A. 3 kilobits and 2 kilobits. B. 2 kilobits and 1 kilobit. C. 2 kilobits, 1.5 kilobits, 1 kilobit, and 0.5 kilobits. D. 2.5 kilobits, 1.5 kilobits, 0.75 kilobits, and 0.25 kilobits. Q.40. Match the cell types listed in Group 1 with associated processes listed in Group 2. Group 1 Group 2. P. NK cells, I. Antibody production. Q. B cells, E. First cells to be recruited at the site of infection. R. Mast cells, E. Antibody-dependent cell-mediated cytotoxicity. S. Neutrophils, I. Histamine production. A. P. E. Q. I. R. 4 statements about lipids are given below as options. Choose the statements which is correct. A. Cholesterol is amphipathic. B. Self-assembly of phospholipids in water is due to hydrophobic effect. C. The temperature at which the gel phase changes to liquid crystalline phase increases with an increase in the degree of unsaturation of fatty acyl tails. D. The choline head group of lipids is positively charged. Q.42. Which of the following techniques can be used to separate proteins according to their molecular weights from a mixture of proteins? A. Ion exchange chromatography. B. Size exclusion chromatography. C. Sodium dodecyl sulfate, polyacrylamide gel electrophoresis SDS page. D. Sucrose density gradient centrifugation. Q.43. B cells produce 2 forms of an immunoglobulin, I. Membrane-bound form, known as B-cell receptor BCR, and E. Soluble form, known as antibody. Which of the following statements is correct about BCR and antibody produced by the same B cell? A. BCR and antibody have identical antigen binding site. B. BCR and antibody recognize different epitopes. C. BCR and antibody are encoded by the same gene. D. BCR and antibody are formed by differential splicing. Q.44. A 100 ml solution of pH 10 was well mixed with a 100 ml solution of. Which one of the following statements on Casparian strips is correct? A. Casparian strips are specific to vascular plants found in epidermal cells. B. Casparian strips are modifications mostly found in shoot tissue. C. Casparian strips act as a cellular barrier to allow selective nutrient uptake and exclusion of pathogens. D. Casparian strips are common in root endodermal cells of non-vascular plants. Q.48. Rotennin is a chemical often used to kill insect pests on crop plants and fishes in lakes. Rotennin acts by inhibiting electron transport from the NADH dehydrogenase enzyme in complex I to ubiquinone in the mitochondrial electron transport chain. Which one of the following explains why plants can tolerate rotennin application? A. The complex I in plants is resistant to rotennin. B. Plants inactivate rotennin by enzymatic degradation. C. Plants have specific channels that efflux rotennin out of the cell. D. Plants have additional NAD/pH dehydrogenases that are resistant to rotennin. Q.49. Although Pseudomonas syringae infection in plants is actively inhibited by the endogenous salicylic acid, SA, of host origin, a successful infection is still established because the bacterium secretes carnitine, an effector molecule. Which one of the following best describes the mode of action of carnitine? A. Carnitine inhibits SA biosynthesis. B. Carnitine promotes the biosynthesis of jasmonic acid, YA, and YA signaling in turn inhibits SA response. C. Carnitine is a structural analog of SA, which binds to the SA receptor and inhibits its function. D. Carnitine is a structural analog of jasmonic acid, YA, which activates YA signaling to inhibit SA response. Q.50. The schematic depicts an unexpanded plant cell within a hypocotyl with the arrangement of cellulose microfibrils marked on its cell wall. Which one of the following shapes would most likely result from the expansion of this cell if the pattern of the cellulose fibrils does not change? A, B, C, D. Q.51. Which one or more of the following statements are not correct with respect to pollen development in angiosperm? A. Tapetal cell wall in all angiosperms breaks down to release the cytoplasmic content. B. Tapetal cell wall in all angiosperms remains intact. C. Tapetal cell wall breaks down in some angiosperm species, whereas it remains intact in others. D. Within an angiosperm species, the tapetal cell wall breaks down in some individuals and not in others. Q.52. Regulation of bisphenol pyruvate carboxylase peptidase governs CO2 fixation in both C4 and CAM-crassulacean acid metabolism plants. Which one or more of the following statements with respect to peptidase activity are correct? A. Peptidase in C4 plants is inactivated by dephosphorylation during the day. B. Peptidase in CAM plants is inactivated by dephosphorylation during the day. C. Peptidase in C4 plants is inactivated by dephosphorylation at night. D. Peptidase in CAM plants is inactivated by dephosphorylation at night. Q.53. Which one or more processes listed below do not produce carbon dioxide during fermentation? A. Brewing wine using yeast. B. Baking bread using yeast. C. Making yogurt using lactobacillus. D. Making cheese using fungus. Q.54. The ovule of a diploid species with 2n=8 undergoes double fertilization. If the pollen is contributed by an individual with meiotic nondisjunction, the chromosome number of the zygote will be blank. XLR, Q55, Q65 carry 2 marks each. Q.55. Match the tasks given in Group 1 with the associated techniques conventionally used as listed in Group 2. Group 1. Group 2. P. Ploidy analysis. 1. RNA sequencing. Q. Profiling DNA methylation. 2. Exome sequencing. R. Identifying non-coding RNAs. 3. Fluorescence in situ hybridization. S. Identifying SNPs. 4. Bisulfite sequencing. T. Satellite DNA isolation. 5. Density gradient centrifugation. A. P2, Q1, R3, S4, T5. Pariderm is a protective tissue found in stems and roots of gymnosperm and woody dicotyledons. It contributes to the increased thickness by secondary growth. Match the paridermal components given in Group 1 with the cell tissue types given in Group 2. Group 1 Group 2. P. Phalloids 1. Tissue resembling cortical parenchyma. Q. Phelogen 2. Cork cambium. R. Phellum 3. Cork-like cells. S. Phalloderm 4. Cork. A. P4, Q3, R1, S2. B. P3, Q2, R4, S1. C. P2, Q. Match the following rice diseases in Group 1 with the causal agents in Group 2. Group 1. P. Blast. Q. False smut. R. Sheath blight. S. Downy mildew. Group 2. 1. Sclerophora macrospora. 2. Rhizoctonia solani. 3. Astelaginoides virens. 4. Piccinia graminis. 5. Magnaporth grisi. A. P5, Q3, R2, S1. B. P4, Q2, R5, S3. C. P4, Q5, R3, S1. Central vascular cylinder or stele consists of the primary vascular system, xylem and phloem, and the associate fundamental tissue. Match the schematics of stele in Group 1, xylem shown in green, and phloem shown as with their respective types in Group 2. Group 1. P. Q. R. S. Group 2. 1. Protostele. 2. Eustele. 3. Atactostele. 4. Ectoploic siphonostellae. 5. Amphiploic siphonostellae. A. P2, Q4, R1, S3. B. P5, Q1, R4, S2. C. P5, Q3, R1, S2. D. P3, Q4, R2, S5. Q.59. Consider the following 4 experimental observations I.e., on the effect of the FORT gene on flowering transition in the shoot apical meristem (SAM) of Arabidopsis thaliana. I. The FT promoter is active in leaves alone. The featuring null mutation causes delayed flowering transition of the SAM. Expressing a recombinant FT protein fused to nuclear localization signal sequence under the endogenous promoter does not rescue the delayed flowering phenotype of the FORT null mutant. Downregulation of FT transcript in the SAM by RNA interference in the wild-type background does not alter flowering transition. Which one of the following conclusions best explains the above observations? A. FT protein resident in leaves causes flowering transition of the SAM. B. FT transcript moves from leaves to the meristem and promotes flowering. C. FT protein moves from leaves to the SAM and promotes flowering. D. Both FT transcript and FORT protein are required in the SAM to promote flowering. Q.60. Which one of the options given correctly matches the alkaloids in Group 1 with their source plants in Group 2? Group 1. Group 2. P. Cocaine. 1. Cocoa. Q. Caffeine. 2. Nightshade. R. Morphine. 3. Coca. S. Atropine. 4. Poppy. A. P3, Q1, R4, S2. B. P-1, Q3, R4, S2. C. P2, Q1, R3, S4. D. Drought-tolerant rice genotype was found to be associated with a missense mutation in the gene A. Which one or more of the following experiments are appropriate to validate whether the mutation in A is the causal factor for drought tolerance? A. Introduce the same mutation in a drought-sensitive rice genotype and test if it becomes drought-tolerant. B. Delete the wild type in drought-sensitive plant and test if it becomes drought-tolerant. C. Determine the stability of the protein encoded by the wild type and the mutant forms of A. D. Repair the mutation in the drought-tolerant rice genotype and test if it becomes drought-sensitive. Q.62. Blue light can directly induce opening of stomata. Blue light also triggers photosynthesis in the guard cells, which indirectly induces stomatal opening. Which one or more of the following experimental approaches would test the direct effect of blue light on stomatal opening? A. Application of low photon fluxes of red light followed by high fluence rate of blue light. B. Application of high fluence rates of red light followed by low photon fluxes of blue light. C. Application of high fluence rates of blue light followed by high photon fluxes of red light. D. Inhibition of photosynthetic electron transport by dichlorophenyldimethylurea (DCMU). Q.63. In a diploid angiosperm species, flower color is regulated by the R gene. Monkeypox is caused by a. Double-stranded DNA virus. B. Single-stranded DNA virus. C. Double-stranded RNA virus. D. Single-stranded RNA virus. Q.67. Which one of the following converts sulfate to hydrogen sulfide? A. Beggiatoa. B. Desulfovibrio. C. Theobacillus. D. Theothrix. Q.68. Which one of the statements about bacterial flagella is correct? A. Flagella varies in length ranging from 0.5 to 2 micrometers. B. Flagella are adjacent fibrils with regular patterns. C. Flagella helps in conjugation. D. Flagella originates from basal body. Q.69. Microbial plastics are made from A. Polyhydroxyalkanoates, B. Polystyrene, C. Polyurethane, D. Polyvinyl chloride. Q.70. The correct sequence of metabolic intermediates in Krebs cycle is A. Alpha ketoglutarate right pointing arrow fumarate right pointing arrow succinate right pointing arrow malate, B. Fumarate right pointing arrow malate right pointing arrow succinate right pointing arrow alpha ketoglutarate, C. Alpha ketoglutarate right pointing arrow succinate right pointing arrow fumarate right pointing arrow malate, D. Succinate right pointing arrow alpha ketoglutarate right pointing arrow malate right pointing arrow fumarate. Q.71. Catabolite repression in bacteria is regulated by the concentration of A. Amino acids, B. Glucose, C. Messenger RNA, D. Lactose. Q.72. Phagocytosis was first described by A. Elie Metchnikoff, B. Robert Hooke, C. Robert Koch, D. Paul Ehrlich. Q.73. Which one of the following statements about batch culture of microbes is not correct? A. Cells from stationary phase will show longer lag phase when inoculated in fresh growth medium compared to those collected from exponential phase. B. Death phase of culture is often exponential in nature. C. Stationary phase is the cryptic growth phase. D. The rate of generation of new cells during exponential growth phase is constant. XLS, Q74 Q84 carry 2 marks each. Q.74. Match the test in Group 1 with its application in Group 2. Group 1. Group 2. P. Oakley-Fulthorpe test. 1. IME detection. Q. Limulus amoebocyte lysate test. 2. Determining antigen-antibody specificity. R. Weil-Felix reaction test. 3. Endotoxin detection. S. Complement fixation test. 4. Rickettsial infection diagnosis. A. P-2, Q3, R4, S1. B. Which one of the following is not correct about antibiotic resistance mechanism in microbes? A. Mycoplasma is naturally resistant to penicillins due to presence of R plasmid. B. Gram-negative bacteria are impermeable to penicillin G. C. Beta-lactamases of bacteria can cleave penicillins. D. Selective microbes can efflux penicillins entering the cell and develop resistance. Q.76. A suspension of photosynthetic green algae was illuminated in the presence of 1,4-CO2 for few seconds. The first metabolite in the Calvin cycle to be radiolabeled will be A. Glyceraldehyde. B. 1,3-bisphosphoglycerate. C. 3-phosphoglycerate. D. Ribulose 1,5-bisphosphate. Q.77. Determine the correctness or otherwise of the following assertion A and the reason are. Assertion A. Endospore can survive heat that would rapidly kill vegetative cells of the same species. Reason R. In endospore, the protoplasm is reduced to minimum volume as a result of the accumulation of calcium dipicolinic acid complexes and small acid-soluble spore proteins, which forms a cytoplasmic gel and a thick cortex. A. Both A and R are true and R is the correct reason for A. B. Both A and R are true and R is not the correct reason for A. C. Both A and R are false. D. A is true but R is false. Q.78. Which one of the following conjugations will result in formation of meridiploids? A. F plus donor times F recipient. B. Ha donor times F recipient. C. F donor times F recipient. D. F plus donor times Ha recipient. Q.79. Which of the following genus is A. Spirochetes? A. Borrelia. B. Leptospira. C. Spirulina. D. Treponema. Q.80. Which of the following is a non-membrane bound inclusion bodies? A. Carboxysomes. B. Cyanophysin granules. C. Polybeta hydroxybutyrate granules. D. Polyphosphate granules. Q.81. Which of the following antibiotics are isolated from Streptomyces spp? A. Gentamicin. B. Nystatin. C. Polymyxins. D. Tetracyclines. Q.82. Which of the following statements about the primary and secondary adaptive immune responses to an antigen is correct? A. IM antibodies appear first in response to the initial exposure of the antigen. B. Majority of the antibodies produced in response to the second exposure of the same antigen are IM isotype. C. Second exposure of the same antigen stimulates production of memory cells. D. Primary antibody response has shorter lag phase than secondary antibody response. Q.83. The spontaneous and induced mutations in bacteria can be distinguished by A. Fluctuation test. B. Replica plating. C. Disk diffusion test. D. Use dilution test. Q.84. During the exponential growth, it took 6 hours for the population. Which one of the following animals has book lungs as a respiratory organ? A. Earthworm. B. Scorpion. C. Octopus. D. Starfish. Q.86. Which one of the following describes the innate behavior of an animal? A. A behavior that is triggered due to the change in environment. B. A behavior that is trained by the parents. C. A behavior that is determined by heredity. D. A behavior that is learned by hit-and-trial approach. Q.87. Which one of the following represents a true ecological population? A. A pitcher plant and a trapped fly in it. B. All animals that live near each other in a national park. C. The leeches and the flatworms that live in a forest. D. All the lions in a reserve forest. Q.88. Which of the following animals show bottle cells during the gastrulation stage of development? A. Snails. B. Amphibians. C. Birds. D. Mammals. Q.89. The organisms that obtain energy from inorganic compounds are known as A. Autotrophs. B. Organotrophs. C. Lithotrophs. D. Phototrophs. Q.90. Which of the following are the causative agents of filariasis? A. Wuchereria bancrofti. B. Leishmania donovani. C. Brugia malayi. D. Trypanosoma gambiense. Q.91. In a population of 1,000 wild dogs in a grassland, 360 and 480 dogs had black body color. Experiment: excess amount of bicadmerna, more than wild-type expression level, was injected into the posterior pole of a wild-type Drosophila embryo at pre-blastodermal stage. Out of the following options, which one represents the best expected phenotype in the resulted developing embryo? A. Normal embryo with head structure at anterior and tail structure at posterior pole. B. Head structure only at posterior pole of the embryo. C. Tail structure at anterior and head structure at posterior poles of the embryo. D. Head structure at both anterior and posterior poles of the embryo. Q.94. Match the hormones precursors listed in column I with the chemical type in column 2 and the tissue of origin listed in column 3. Column I. Column 2. Column 3. P. Glucagon. I. Tryptophan derivative. A. Anterior pituitary. Q. Pregnenolone. E. Peptide. B. Pineal. R. FSH. E. Steroid. C. Adrenal. S. Melatonin. I. Glycoprotein. D. Pancreas. A. P E D Q E C R I V A S I B. B. Match the syndromes listed in column I with the cause symptoms listed in column 2. Column I. Column 2. P. Prader-Willi syndrome. I. A collection of signs and symptoms due to prolonged exposure to corticosteroids like cortisol. Q. Down syndrome. E. A syndrome of inadequate reabsorption in the proximal renal tubule of the kidney. R. Cushing syndrome. E. A genetic disorder usually caused by the deletion of a part of chromosome 15. S. Turner syndrome. I. A genetic disorder caused by the presence of all or part of a third copy of chromosome 21. T. Fanconi syndrome. V. A genetic condition in which a female has partially or completely missing an X chromosome. A. P. E. Q. Match the immunological statements in column I with the appropriate descriptions from column 2. Column I. Column 2. P. Active acquired immunity. I. Complement proteins and interferons. Q. First line of defense. E. Direct contact with pathogens that enter the body. R. Passive natural immunity. E. Surface barriers. S. Second line of defense. I. Antibodies pass through placenta. A. P E Q E R I V S I. B. P I V Q E R I S E. C. P I V Q E R E S I. D. P I Q I V R E S E. Q.97. Match the standard stated cofactors in column I with their respective enzymes in column 2. Column I. Column 2. P. Cu2+. I. Dinitrogenase. Q. She. E. Cytochrome oxidase. R. Ni2+. E. Pyruvate kinase. S. K+. I. Glutathione peroxidase. T. Mo. V. Urease. A. P. VQE R I V S I T E. B. P E Q I V R V S E T I. C. P I V Q E R. Presence of excess glucose has been known to prevent the induction of lac operon as well as other operon-controlling enzymes involved in carbohydrate metabolism in E. coli. Which of the following processes defines the phenomenon? A. Catabolate repression. B. Attenuation. C. Glucose effect. D. Feedback inhibition. Q.99. Which of the following techniques is used for determining the three-dimensional structure of proteins? A. Cryo-electron microscopy. B. Circular dichroism. C. Nuclear magnetic resonance spectroscopy. D. X-ray diffraction. Q.100. Among the following statements, which is true regarding the replication of DNA? A. Replication is bidirectional and conservative in nature. B. Replication in eukaryotes takes place at multiple ORI sites simultaneously. C. Both the strands replicate in discontinuous manner. D. One strand replicates in continuous while the other replicates in discontinuous manner. Q.101. Which of the following statements is true for colchicine? A. It binds to tubulin molecule and disrupts the assembly polymerization of microtubule. B. It inhibits crossover of chromosomes during meiosis. C. It inhibits chromosome condensation during prophase. D. It blocks mitotic cells in metaphase. Q.102. Wild-type Drosophila females having 3 link genes AABBCC were crossed with triple recessive mutant ABCC males. The F1 female. Choose the correct group of fat-soluble vitamins. A. Cholecalciferol, alpha-tocopherol, menadione. B. Thiamine, cholecalciferol, alpha-tocopherol. C. Niacin, alpha-tocopherol, menadione. D. Biotin, thiamine, niacin. Q.105. The synthesis of thyroxine T4 in human body requires A. Selenium, B. Iodine, C. Iron, D. Zinc. Q.106. Which among the followings is not an essential amino acid? A. L-phenylalanine, B. L-valine, C. L-lysine, D. L-arginine. Q.107. The time required for stipulated destruction of a microbial population at a given temperature is A. D value, B. F value, C. Z value, D. Q10 value. Q.108. Which among the following statements is not correct? A. Codfish is a major source of omega-3 fatty acids. B. Beetroot is a good source of beta-carotene. C. Apple is a good source of vitamin B12. D. Fresh sugarcane juice is a good source of polyphenol oxidase. Some of the industrial products are produced by fermentation processes. Identify the correct pair of product and fermentative microorganism. A. Vinegar, Acetobacter aceti. B. Citric acid, Enterobacter aerogenes. C. Ethanol, Saccharomyces cerevisiae. D. L-lysine, Aspergillus niger. Q.113. Choose the correct statements about the enzyme and its application in food processing reaction. A. Chymosin is widely used in cheese manufacturing. B. Thymolysin is used in the synthesis of aspartame. C. Beta-galactosidase catalyzes the hydrolysis of galactose. D. Lipase is used for restructuring of acylglycerol. Q.114. Identify the Gram+V bacteria responsible for causing food-borne diseases among the followings. A. Campylobacter jejuni. B. Clostridium botulinum. C. Vibrio cholerae. D. Salmonella typhi. Q.115. Extrusion cooking is accomplished in 4 different stages, which are indicated as I, II, III and IV in the figure given below. Choose the correct option representing the name of each stage. E. Temperature, pressure. IV. E. I. Extrusion cooking stages. A. I. Feeding. 2. Cooking. 3. Kneading. IV. Expansion. B. I. Kneading. 2. Feeding. 3. Cooking. IV. Expansion. C. I. Feeding. 2. Kneading. 3. Cooking. IV. Expansion. D. I. Cooking. 2. Kneading. 3. Feeding. IV. Expansion. Q.116. Match the method value used for measuring lipid characteristics in column I with the corresponding properties indicated by them in column 2. Column I. Column 2. P. Thiobarbituric acid test. 1. Induction time. Q. Ransomat method. 2. Degree of unsaturation. R. Peroxide value. 3. Carbonyl content. S. Iodine value. 4. Hydroperoxide content. A. P-3, Q1, R4, S2. B. P-1, Q3. Match the peeling technique in column I with the vegetable, for which it is used in industry, given in column 2. Column I. Column 2. P. Knife peeling. 1. Brinjal. Q. Abrasion peeling. 2. Tomato. R. Flame peeling. 3. Potato. S. Flash peeling. 4. Cucumber. A. P-3, Q4, R1, S2. B. P-4, Q1, R3, S2. C. Match the process in column I with the related food component in column 2. Column I. Column 2. P. Caramelization. 1. Lipid. Q. Denaturation. 2. Sugar. R. Oxidation. 3. Pigment. S. Bleaching. 4. Enzyme A. P-2, Q4, R1, S3. B. P-2. Identify the correct statements related to grain polysaccharides among the followings. A. Dextrin are a group of low molecular weight polysaccharides produced by dry hydrolysis of starch. B. Amylose is a linear polymer of D-glucose units joined by alpha-1 right-pointing arrow 6 glycoside linkages. C. Amylopectin is a branch-chain polymer of D-galactose monomer units. D. Retrogradation is a process of reassociation of amylose and formation of crystalline structure by gelatinized starch upon cooling. Q.120